want to talk about today is a role for functional assays in precision oncology trials. And so where I'd like to start is thinking about when we talk about precision medicine these days, what we're generally talking about by and large is genomics. We talk about sequencing a patient's cancer, identifying potential targetable mutations, and then trying to treat those patients with specific drugs that are targeted toward those mutations. And this is it's an interesting approach. It's been successful in many cases, but to be quite honest with you, there are really too many mutations to be able to drug at the present time. And really the majority of patients have such complex and heterogeneous cancers that you really have multiple mutations, and often the problem is you don't know which one is actually a driver mutation and which are the ones that are just passengers. And so when you sort of when you use a genomics approach, you're really sort of again picking sort of an option just because there's a drug that targets that particular mutation, but you don't really know whether or not the patient's cancer is being driven by that particular mutation or being modified by other mutations. And so this is an article by Tony Latai a few years ago. He really pioneered BH3 profiling and functional assays, where he makes the case that what we really want to do is, in addition to a genomics approach, we want to bring in other modalities to be able to test cancer therapies. And so what he suggests is that really what we want to do is take a comprehensive approach in precision medicine, where you take a patient's tumor and really profile it in a multi-omics approach, where it's not just DNA mutations, but you also look at proteins, RNA expression, as well as the epigenome, in order to fully understand what's happening with regard to the biology within that patient. In addition, you want to always use your, make your treatment decisions based on clinical experience and what's in the literature. And what he also makes the case is that what we want to do is be able to interrogate the tumor cell itself and really determine whether or not there are specific drugs that particular tumor may be sensitive to. And that's what he refers to as functional assays. And then all of this information should be collated, gathered together, and presented to a tumor board, where there's a multidisciplinary approach to really individualize cancer therapy for patients. And that's really what we've taken on is this functional assay in hematologic cancers is where we're starting, and then we're starting right now to start layering on all of the other information that comes from these functional assays in specific patient samples, so that we can really understand the biology within individual cancers. So what we're doing is using flow cytometry to really take a look at a patient's blood cancer and really try and understand what components of a patient's bone marrow or peripheral blood may be sensitive to different agents. And so what we do is have a high throughput flow cytometer, where a patient's blood cells are actually put through the scanner in sort of a single cell matrix, where a laser is focused to scatter light, and each of the cells that go through this laser have already been labeled with a fluorescent tag for a different cell surface protein, so that it marks the different types of cells that are present in that sample. And so how we do this is again, we take the patient sample and put it through the flow cytometer, where we'll be able to actually take a look at the live cells, and using those different cell surface markers, exclude the different mature lineages, such as myeloid cells, monocytes, lymphocytes, erythroblasts, and granulocytes, and really get down to what we're considered the lineage negative cells, which are the undifferentiated cells. And within this population is where you do see, take some additional markers to eliminate other differentiated cells. And then this is the total blast population is what you're left with. So for example, in a myeloid disease like AML or MDS, you can use a process like this to really hone in on the cancer cells within the patient sample and really determine at the single cell level whether or not a drug is affecting that population only, or if there are other populations that may or may not be uh, affected as well. So what we do is actually take our platform and test drugs directly on patient samples. And so what we'll do is get a bone marrow from a patient that's been collected overnight and shipped to us. And what we do here is just a simple red cell lysis, so that we remove all of the red cells from the sample, and what we're left with is a population of leukocytes, which include normal peripheral blood or bone marrow components, in addition to the malignant cells. And then what we can do is split that sample into multiple aliquots in 384 well plates, and then test each of those wells with different drugs and drug combinations. So, for example, if we were to test single doses of drugs and drug combinations, we can actually, on a single patient sample, test hundreds of monotherapies as well as combinations to really identify what is the profile for that patient in terms of drug sensitivity. We can also go to the other end of the spectrum, where we test, for example, 3 specific drugs in multiple dose responses and in combination, so that we can really understand what a patient's sensitivity is to a specific drug. And then what we have is an automated analysis pipeline that allows us to produce these results in about 7 to 14 days. And so I mentioned that we work primarily in blood cancers, and we're starting first with a disease called high-risk myelodysplastic syndrome. It's actually a rare disease where there are only about 12 to 20,000 patients per year. It affects primarily an older population of patients, and really it's a high-risk disease because 1 out of every 3 MDS patients actually progress to acute myeloid leukemia, which is a deadly disease. And the average lifespan of a patient diagnosed with myelodysplastic syndrome is about 6 years. But there are certain high-risk categories where survival is actually very low and is measured in months. And if you take a look at the genomic landscape of MDS, it's actually highly complex. If you look at all of these different mutations here, these are on the y-axis are the different mutations that are present, that have been identified in 944 different patient samples, and across the x-axis is all 944 patients. And you can really see that while there are certain mutations that tend to be common across many different samples, again, as you get out toward the tail, it becomes, again, a very, very complicated disease. So really what happens is you wonder to yourself, how do you treat myelodysplastic syndrome in a precision medicine framework where you're trying to target a specific mutation? So what we've done is performed an initial pilot study with Stanford University and Dr. Peter Greenberg, where we profiled the bone marrow aspirates of about 56 different MDS patients with high-risk MDS. And what we did was we tested up to 76 different approved and investigational therapies, not necessarily for MDS, but they could be, for example, things that you wouldn't normally consider treatments for blood cancers, such as calcitriol or olaparib, which is a PARP inhibitor, or even these CDK4/6 inhibitors. And so what we've done is identified by screening in sort of this broad agnostic way that we can define and cluster patients with high-risk MDS into really three classes, where there's this what appears to be this more chemo-resistant phenotype, or a more chemosensitive phenotype, and then here's another that seemed to be more sensitive to some interesting agents, such as some differentiating agents, some hypomethylating agents, and even some of the some drugs that you wouldn't normally consider treating a blood cancer with. And so the question becomes, here is now a list of drugs that probably should be studied in MDS, because we can potentially identify subpopulations of patients that would benefit from a particular therapy. But then, how do you study so many drugs in such a rare disease? And so, if you look at the traditional randomized controlled trials, what will happen is, again, everybody knows this. Most patients are randomized to a control group or to drug A, and you look to determine whether or not the drug A is better than the control. But what happens if drug A is only effective in 10% of the patients, and there's no way to select those patients? And if you do this across multiple different drugs, you can see that it becomes a factorial problem that becomes very problematic in a rare disease. So one potential solution is actually to do a platform trial in which you take multiple drugs within the same clinical trial architecture and compare them against a common control group, so that patients are randomized in a fixed ratio to either a control or to each of the different drugs that are on the trial, so that you can efficiently use patients in a rare population to study as many drugs as possible. And one of the pioneers of this approach is Don Barry, who is a biostatistician at MD Anderson. And what he's proposed on top of these platform trials is actually to use Bayesian adaptive randomization, in which you will enroll a patient and you'll randomize them and you'll record the outcome to the multiple therapies. And then, based on the outcome or treatment responses in the patients who've already been enrolled, what you do is you update the trial data and you update the statistical model, so that drugs that are better within a particular population can be quickly identified. In addition to identifying drugs that are ineffective in that population, so that you can actually get rid of drugs relatively quickly that are ineffective in that disease, and then you update the randomization weights, and then continue this process by enrolling more patients. And so this is a process that's actually been used very successfully. For example, in the I-SPY 2 trial, where this has been shown in neoadjuvant breast cancer, where you have multiple investigative agents that are combined with paclitaxel. The end readout of the study is actually a pathologic complete response. And so what they've managed to do over 10 years is identify very quickly drugs that are very effective in breast cancer in the setting, and also quickly eliminate drugs that are ineffective. And so here, over the last 10 years, you can see that the number of drugs and drug combinations where a number have actually been approved relatively quickly using this approach, and have also halted a number of ineffective drugs that were being developed in breast cancer. And so we want to use this as a paradigm for how to move forward. One of the most recent Bayesian adaptive platform trials is GBM Agile. And what you can see here is in another disease like glioblastoma multiforme, where no drugs have been really successful over the last several decades. You can compare multiple drugs under the same conditions and really identify effective therapies and quickly fail ineffective therapies. And again, this is also being done in pancreatic cancer, where there's a common theme here between GBM Agile and Precision Promise. Is we've been very unsuccessful over the years in identifying therapies that are effective. And so, really, the advantages of a Bayesian adaptive platform trial like this is that there's a path to an accelerated approval by FDA, as also in addition to fast failure. And again, all of these things you can potentially accelerate approval, reduce costs since you're sharing trial infrastructure and a common control group. You can potentially increase enrollment because you're at centers of excellence that have access to these patients, and improve the understanding because every single patient is contributing to the overall knowledge of the clinical trial as it's being conducted. So what we're envisioning is doing a similar trial in myelodysplastic syndrome, where a patient will be enrolled and consented. We'll obtain a patient sample, so in this case it will be a bone marrow sample, and we'll do a predictive biomarker test, which is the notable test for drug sensitivity. That information will be fed into the randomization system so that let me see if I can get the laser. I can't get the laser going, but that the information from the drug sensitivity test will be fed into the randomization system, so that the information regarding a patient's sensitivity to drug A, drug B, or drug C is used to weight their probability of being randomized to a drug where they're more likely to respond than not. So the trial architecture that we're building is at first there'll be a stage one where we are looking at complete response, which is a surrogate endpoint in the trial, where, for example, multiple drugs can be in different stages. So, for example, drug A could be in an early phase of the study where it's just entered, after the trial's been running for a period of time. Drug B could be a drug that's been in the trial for a while and has actually shown sufficient efficacy that there's a high probability that it's going to beat the control group in a defined population, such that it's now graduated to a pivotal cohort. And drug C is again in the middle of this trial, where we're still learning how to use the drug sensitivity score to predict the response. And in all cases, there is a concomitant control group or a common control group where a small number of patients are being enrolled, so that we actually have a comparison, so that we can identify. Once this signature has been identified and the drug has been graduated to a pivotal cohort, that it's actually a predictive biomarker rather than a prognostic biomarker. Because if it's a prognostic biomarker, you should see no difference in that in treating a patient with drug B versus the control. If it's a predictive biomarker, certainly you should be able to ascertain that drug B is more effective than the control. And our goal here is really to get both the drug registered and the device registered as a predictive test at the end of this stage 2. And so what I want to do is just show you a quick simulation that our engineers built, where what we've done is taken three different drugs: drug A, drug B, drug C. On the left-hand side is the notable platform, which uses the drug responses to randomize patients to the different arms of the clinical trial. On the right-hand side, this is a standard Bayesian adaptive platform trial in which you have randomization probabilities based on how well each of these drugs is performing. And what I want you to get from this is that for drug A, the underlying response rate is about 10%. For drug B, we're assuming that the underlying response rate is about 20%, and drug C, the underlying response rate for the drug is about 30% in the overall population. And so you can see at the very beginning, on the right-hand side, for drug A, the randomization probabilities are equal because we have no information about treatment responses. And so it's a third, a third, a third. And so if we add 10 patients, what we'll do is, so there are 10 patients over here, 10 patients over here, and we'll simulate one patient. And so on the right-hand side, this patient is just randomized, again, one to any of the three cohorts. And the color here indicates that the patient's drug sensitivity is actually to drug A, but the patient was randomized to drug C. Whereas on the right, on the left-hand side, using the notable test, the patient is properly randomized to drug A. And so if you do this multiple times, again, here's another patient who's most sensitive to drug A and was assigned to drug A. Here's a patient who was, again, same patient, sensitive to drug A, but was now put into drug B, into arm B. Next patient, here's a patient whose drug sensitivity is sort of equivalent all the way across, but that patient over here gets randomized to drug A. And so if we keep doing this over and over again until all 10 patients are randomized, okay, and then put them down here. So green are true responders, red are the non-responders. And so you can see on the left-hand side, using drug response data, that 4 out of 5 of the patients who were randomized to drug A were actually responders. There were no patients randomized to drug B, and to drug C, there were 4 out of 5 were responders. On the other side, there were 4 patients randomized to drug A. There were no responders in that group. 2 out of 6 patients were responders to drug B. And so you can see the randomization probabilities on the right-hand side now are 16%, 48%, and 36%, reflecting the results of the clinical trial. So if we continue to do this over multiple iterations, and do this and add up to 100 patients. Okay. Okay, so now we're at 100 patients, and you can see on the right-hand side in a standard adaptive platform trial. So for drug A, you see that 10 out of 29 patients were properly were responders to drug A, and the current randomization probability is 36%. For drug B, only 4 out of 19 patients were actually responders, and so the randomization probability for drug B is 15%. For drug C, 20 out of 52 patients were responders, and so the randomization probability is 50%. So this is in many ways a sort of a pick the winner type of approach. Whereas on the left-hand side, if you use drug sensitivity as a way to identify patients and put them into various cohorts, for drug B, you can see that actually there were 27 responders out of 38 in drug A, 13 out of 19 in drug B, and 23 out of 43 for drug C. And the overall response rate across the entire trial is 63% when you use drug sensitivity as a predictor of response and use it in the randomization algorithm. Whereas on the right-hand side, the overall response rate is 34%. So our contention is that you can use drug sensitivity data, the functional assays, and incorporate them into precision oncology trials so that you can benefit patients by putting them into arms in which they're more likely to respond, and benefit the therapeutics companies by identifying the subpopulations of patients that are most likely to respond to their therapy and accelerate their path to approval, because you can do this with smaller sample sizes in a much quicker period of time in a less expensive platform format that uses a common control rather than a single randomized controlled trial. So our regulatory path for this right now is, we're likely to go and try and apply for a single site PMA. We actually have an excellent internal team that is working to develop specific drug tests and get them into our CLIA lab so that we can start to use these as part of a investigational device exemption for a clinical trial. And we have an external team of consultants that we're using to really move this forward with the regulators. And so actually on Monday, we have our first Q-Sub meeting with CDRH, really to get agreement on what are the analytical validation studies we need to do for flow cytometry, as well as how we get feedback on how we plan to use the output from the assays to predict responses. And then early next year, we're going to approach CDER, the drug side of FDA, in order to get agreement on the master protocol design, what our endpoints will be, the control therapy, and the adaptive randomization algorithm, as well as criteria for registration, as well as potential accelerated approvals. We have a number of key partnerships. We've been working with the Global Coalition for Adaptive Research, who's has been advisors to us for trial operations and is managing our CRO selection process. The MDS Foundation, who is they're a terrific international organization in which there are patient advocates who are looking at our clinical trial and helping us with how to incorporate not only PROs but also get patient input into how do we explain a complex clinical trial like this to MDS patients, and why is it valuable to them to enter the trial. And again, we're working with Berry consultants to work on all of the simulations. And then these are the various KOLs who've been reviewing our protocols and really providing valuable input to the overall strategy of executing this clinical trial. And with that, I'll thank you very much and take any questions.
Thank you very much, Dr. Hiroomi Tada, for your presentation. The floor is now open for any questions for Dr. Tada.
It's very fascinating, and I worked at BD Biosciences in San Jose, and so a number of years ago, so looked at some of these kinds of applications of flow cytometry. One of my questions is this either now or in the future potentially applicable to looking at the drug sensitivity of cancer stem cells?
So it depends on the limit of detection and how high how great the population of cancer stem cells are. If it's a relatively rare population, the limit of detection for flow cytometry is it may not be sensitive enough to be able to identify those rare populations. What we hope to be able to do is, over time, if we get serial samples on patients, really look at clonal evolution and what's happening to those patients under selective pressure from various different therapies. And so we actually have an observational trial ongoing right now where we're collecting serial samples from patients who are on standard therapies, so that we can at least try and understand what's happening to the different clones that we identify at baseline, or the different mutations that we identify at baseline, and how their cancer evolves over time, under conditions of various therapies. That's the best we can do until we can get a flow cytometry or some other assay that allows us to really get down to this much more single cell level. Much like what Mission Bio is doing with their single cell sequencing.
Right. And in a similar vein, could it be applicable to patients that are MRD positive?
So again, it really depends on the limit of detection, and so we are working on some MRD assays right now, but it's been tough. Even with the 3-color laser, you're limited to about 11 different cell surface markers.
Right.
We're actually pivoting to a 5 laser
Oh.
instrument, which will let us go up to 25 different markers, and so that may also help us in terms of identifying some of these rare populations where you need 2 or 3 different 3 or 4 different markers on a cell to really differentiate them from other types of cells.
And my last question, it might be premature because you mentioned you're going to meet with CDRH on the 19th, but I'm just curious about the path to PMA. In other words, do you look at patients managed, kind of like in your simulation, managed via this versus historical controls of patients who've been managed conventionally or even in a non-notable adaptive design? Do you have thoughts about that, or is that something that you're working out?
I think we're in the process of working it out. I mean, I think the one area that a test like this can really help is, for example, in multiple myeloma, where you have multiple therapies that have been approved recently in sort of the second, third, fourth, fifth line setting, and physicians actually have no idea how to use any particular drug for multiple myeloma patients at any given time. The physicians are sort of working out algorithms in terms of sequencing and things, but a lot of that is really quite empirical. And so, what we're imagining is in the future, if there are multiple drugs that have been approved with a diagnostic test like this, that you can run a single sample across multiple different drugs and really try and come up with the best therapeutic option for a particular patient at that particular time.
Fascinating. Thank you.
Yep. Thank you.
Thank you, Dr. Hiroomi Tada.